Hello, everybody, and welcome to this breakout session hosted by ProBio, one of the four sponsors in today's virtual summit for the state of cell and gene therapy. I'm Kevin Davis. In this session, G ProBio's Associate Director of Product Marketing, is going to talk to you about the in vivo CAR T revolution, insights and impacts on developers. Here then, without further ado, is G from ProBio.
Thank you for your introduction. Today, we are going to talk about navigating in vivo CAR T development, overcoming CMC hurdles from bench to bedside. Here are today's presentation topics. First of all, we are going to walk you through in vivo CAR T market landscape. Then we will talk about the CMC challenge and solutions in mRNA tLNP and LVV drug product. In the last part, I will introduce ProBio's CDMO service offering and how we accelerate your in vivo CAR T discovery and development. Okay, so let's move to the market landscape of in vivo CAR T. CAR T are powerful therapeutic agent for blood cancer and have the potential to revolutionize therapy for many other diseases. To circumvent the complexity and high cost, alternative strategies to generate CAR T cell in vivo have received a great deal of attention in the past few years. The chart on the left shows the ex vivo CAR T production process. A patient's T cells are collected and then shipped to a lab, genetically modified to carry the CAR and grow into large numbers. After QC release, they are shipped back to the hospital for infusion. The entire one-to-one process takes 2 to 7 weeks, during which a patient's disease may progress. The chart on the right shows the in vivo CAR T manufacturing process, which aims to be an off-the-shelf treatment. CAR therapy as nanoparticle formulated with mRNA or engineered viral vectors is manufactured in large and ready-to-use batches. When a patient needs treatment, this product is infused directly. The product then instructs the patient's own T cell inside the human body to become CAR T cell, eliminating the need for the external cell manufacturing and the long wait. The in vivo CAR T holds transformative potential for the treatment of cancer and a range of diseases. In addition to that, it could streamline manufacturing, simplify logistics, lower cost, enhance the biology, and also improve the safety, especially in the tLNP scenario. However, it can also introduce distinct challenge in delivery and expression. The genetic cargo must be delivered efficiently and specifically to the intended target cells. Off-target delivery can compromise efficacy by diluting the therapies. And if the cancer cells are accidentally modified, they may also hide the target antigen, preventing T cell from attacking them. There are two major approaches, viral and non-viral delivery, in the field of in vivo CAR T. The slide shows the comparison. On the left, it is LNP-mediated transient CAR expression, also known as mRNA tLNP. T means targeted. In this approach, an anti-CD3 antibody targets the LNP to the T cell. After binding, the LNP enters the cell while endocytosis. Inside the endosome, it releases mRNA coding CAR. The mRNA is then translated into CAR protein, leading to a temporary CAR expression and transient T cell redirection. This approach has several advantages. It can be produced in a cell-free manner, minimizing variability of the product. What's more, mRNA administration poses no risk of insertional mutagenesis as the cargo is delivered and translated to the cytoplasm. On the right, it is the viral vector-mediated permanent CAR expression. In this approach, a lentiviral vector is engineered with an anti-CD3 antibody for T-cell retargeting. Upon binding, the virus enters the cell and its genetic cargo is reverse transcribed and permanently integrated into the host genome. The CAR gene is then expressed continuously, resulting in stable genetic modification. As LVV has now been widely used for therapeutic applications, the manufacturing process and reagent are well established and validated. However, LVV's ability to cause the permanent genetic modification may pose risk in safety. Specifically, accidental transduction of germ cells must be absolutely prevented. The advancement of in vivo CAR T therapy has become a significant highlight in the industry since 2024. Many pharmaceutical companies are proactively establishing in vivo CAR T platform with nearly 10 major collaborations in this field between 2024 and 2025. These transactions include technology platform licensing, joint development, company acquisition, etc. Driven by this momentum, capital investment has surged and R&D pipelines are expanding rapidly. Currently, over 100 biotech companies specializing in cell therapy and mRNA technology have entered this field. We anticipate that with the next 12 months, many projects will advance to the stage of IIT or IND application. In some well-known in vivo CAR T BD transaction, the compelling early human data of investigator-initiated trial, in short, IIT, have played an important role. Among the major markets, China offers a unique environment for IIT in parallel with industry-sponsored trial for CGT product. In IIT studies, investigators can generate first-in-human safety and efficacy data quickly at top-tier hospital before or parallel to the full IND filing. The findings from IIT study not only serve to validate the technology platform and the pipelines, but also generate critical data package to support IND filing and other regulatory milestones. Importantly, these results can significantly enhance a CGT company's position in securing funding, advancing towards an IPO, or negotiating out-licensing agreement. Moreover, by enabling the execution of high-quality CGP-compliant clinical study at less than half of the cost of a comparable U.S. trial, IIT study also meaningfully extends a CGT company's cash runway and preserve capital for key development priorities. We believe IIT will be an important regulatory pathway to enable and accelerate in vivo CAR T technology and pipeline development. As a brand new field, speed to IIT or IND translates directly to the value and competitiveness. However, there are a lot of challenges in CMC which may delay IIT filing or IND filing. In the next part, I will dive into the CMC challenge and the solutions in mRNA tLNP. mRNA tLNP is a nanoparticle that are engineered with surface ligand, most commonly antibody fragment, to direct them to the specific cells. As a novel therapeutic modalities, mRNA tLNP presents several distinct CMC challenges. First of all, the targeting antibody are typically antibody fragments, which bring inherent CMC difficulties, including low expression titer and complex purification requirements. Secondly, scaling up from laboratory scale to clinical or commercial scale presents significant technical hurdles. Thirdly, the ratio of antibody to LNP must be carefully optimized for each candidate, and it directly impacts both in vitro and in vivo efficacy. Finally, it currently lacks mature and standardized analytical method, requiring extensive in-house development. To address those technical challenges, ProBio has established a set of technology platform and a toolbox tailored to mRNA tLNP development. So first of all, to address the challenge of targeting antibody, ProBio has established a CMC platform that supports various antibody fragment formats, including VHH, scFv, and Fab. Our platform enables rapid cell line development, delivering the top 6 clone with 10 weeks from DNA synthesis, with expression titer reaching up to 50 grams per liter. We have also implemented template-based purification and analytical methods specifically tailored to each fragment format, ensuring accelerated development timeline and scalable GMP production. We have successfully delivered or are currently advancing 11 antibody fragment CMC project, and the most advanced VHH-based project has achieved IND clearance in the US. To address the scalability challenge for mRNA tLNP, ProBio has established a versatile system which are compatible with a range of LNPs, RNA, and antibody formats. Our streamlined manufacturing process achieves over 95% of LNP modification rate and over 60% of total mRNA recovery rate from small process development scale to gram-level GMP manufacturing. At ProBio, we provide 3 conjugation approaches for tLNP development. Surface conjugation via Sire and maleimide chemistry, which is a covalent method offering a robust, consistent, and well-characterized process. Surface conjugation via Dipical and azide chemistry, which is also a covalent approach, delivering strong bonding and process reliability, while it requires stringent control over residue azide. Post-insertion method, which is a flexible, no-covalent strategy suitable for rapid research and screening with higher product variability, making it less ideal for GMP manufacturing. Each method has its advantages and limitations, so our team will work closely with you to select the optimal strategy based on the development stage, molecular, and final product requirements. For mRNA tLNP, the antibody to LNP ratio must be carefully optimized for each candidate molecule, as it critically influences both in vitro and in vivo efficacy. It is important to recognize that higher conjugation density does not always translate to better performance. In some cases, over-conjugation can alter antibody's spatial conformation, reducing its ability to binding the T cell and potentially compromising tLNP stabilities. For each specific project, through systematic adjustment of reactive component content in LNP formulation, which impact LNP surface active site, and also the adjustment of quantity of reduced antibody during conjugation reactions. we identify optimal LNP formulation and conjugation conditions guided by cell-based assay. As shown in these two figures, LNP formulation 2 was identified as optimal formulation, while conjugation condition 3 was further selected as the ideal antibody to LNP ratio. To support process development and product release, ProBio also established a comprehensive and robust mRNA tLNP analytical platform. Building on our standard LNP analytical platform, this new platform has been further enhanced with dedicated method for comprehensive characterization of tLNP product, including antibody content, biological activity, and conjugation density, etc. These slides show you some example of tLNP characterization. Cryo-EM image confirms tLNP integrities. ELISA method is developed to measure LNP surface antibody conjugate concentration, and nanoflow is developed to quantify antibody molecular on LNP surface. Functionality is validated in cell-based assay using YORCAT and PBMC. In addition to the technical challenge, complexity in project management is another challenge in tLNP project. In this kind of project, customer usually needs to work with multiple partners for plasmid, antibody, mRNA, LNP. This causes delay, extra cost, and complexity. ProBio simplify everything with a seamless integrated CMC strategy, consolidating all. So one partner from idea to IND. With this truly one-stop mRNA tLNP CMC solution, we are confident to offer 6 months from DNA to IIT-ready material and 9 months from sequence to IND-ready material. In the third part of my presentation, I will dive into the CMC challenge and solutions in vivo LVV CMC. Developing lentiviral vector for in vivo CAR-T, known as LVV drug product, presents a unique set of technical and analytical challenges. In the upstream process development, crude titers are often reduced due to the engineering of VSVG envelope plasmid and the introduction of additional plasmids encoding the targeting antibodies. This modification can also result in lower overall transduction efficiency. In the downstream process development, vector aggregation becomes a common concern as the native envelope proteins are modified. It also requires more extensive characterization and stricter control for safety and impurity profiles, such as HCP. Finally, tailored analytical method for LVV drug product must be developed. So to meet these demands, comprehensive and reliable in-house analytical platform are essential. In ProBio, we have established IIT IND enabling CMC solutions tailored for LVV drug product. So first of all, in LVV drug product, the envelope plasmid are usually divided into 2 functional parts, binder and fusogen. So 4 to 7 plasmid are usually used in LVV packaging. In ProBio, we perform customized plasmid ratio optimization, especially for binder to fusogen ratio, to maximize LVV transduction. In the upstream process development, we improve the crude titer by optimization of transfer plasmid and upstream process parameters. Furthermore, to address the downstream challenge, we tailor our downstream purification and formulation development to each specific envelope, ensuring high recovery and stability. Moreover, to deliver a complete data package for regulatory submission, ProBio has a ready-to-use LVV drug product analytical platform, especially for potency and safety profile, and also the drug product-related physicochemical profile. This slide shows ProBio's in-house analytical platform for LVV drug product. In addition to the quality attribute that we usually studied in ex vivo CAR-T, we also developed the additional method for biological activity, visible particulate matter, etc. This dedicated platform ensures that every batch of LVV drug product is thoroughly characterized, supporting both patient safety and regulatory success. For LVV-based in vivo CAR-T, we are confident to offer DNA to IIT-ready LVV in 4 months and DNA to IND-ready LVV in 9 months. In the last part of my presentation, I would like to give you a quick introduction of ProBio and our service offering in vivo CAR-T. ProBio aims to be a leading global CDMO, renowned for cutting-edge technology to enable our customer to deliver life-changing therapies. ProBio provides end-to-end CDMO service for cell and gene therapy, mRNA, and biologics, supporting customers from drug discovery through development to the clinical and commercial manufacturing. At ProBio, we have built up a strategic global network to deliver resilience, flexibility, and scalability for our partners worldwide. For cell and gene therapy, we provide integrated process development and GMP manufacturing across the US and China. Our facility in Hopewell, New Jersey, specializes in plasmid and viral vector development and manufacturing, serving as a key supply hub for the US and the European markets. Meanwhile, our large-scale plasmid, viral vector, and mRNA production facility in China offer cost-effective, high-volume manufacturing capacity, supporting global supply chain, IIT material supply, and the rapidly expanding APAC region. To meet the growing customer needs in vivo CAR-T, ProBio has integrated its service platform in discovery, pharmacology, plasmid, biologics, LVV, and mRNA LNP to rapidly establish a discovery to IND solutions covering LVV drug product and mRNA-targeted LNP. In the field of in vivo CAR-T and in vivo gene editing, we have partnered with 8 customers across the world for the development and manufacturing for IIT or IND purpose. The most advanced projects are in the phase 2 development in the US and China. To conclude my presentation today, ProBio is committed to providing the innovative and integrated discovery to IND solutions to bring your in vivo CAR-T therapies to the patient faster. Okay, thank you for listening.
Thank you, Chi, for that very nice presentation, and thanks to ProBio for their generous support of today's summit, The State of Cell and Gene Therapy. Coming up in our next session, Jennifer Adair from UMass Chan Medical School. She'll be presenting on strategies for expanding access to cell and gene therapies around the world. See you in the next session.